Today, many cancer drugs are designed to work only in patients whose tumors have a specific molecular biomarker. These personalized medicine approaches depend on knowing a patient's biomarker status. But in reality, biomarker testing isn't yet routine in the clinic. Most patient samples are collected to confirm a diagnosis, not to identify molecular features. So when a researcher goes to a biobank looking for lung cancer samples with a specific biomarker, the answer used to be, we have the lung cancer samples, but we don't know which samples actually carry that specific mutation. The only way to find out was to do sequencing of many biospecimen samples to find the ones of interest for a given project. That gap is what historically made biomarker-driven research slow and expensive. From tissue to biomarker, the benefits of pre-characterized biospecimens for precision oncology. When a biobank does not provide molecular data, researchers are forced to bridge the gap between biospecimens and sequencing on their own. This typically leaves them with two options: generating sequencing data in-house or outsourcing to a third-party sequencing vendor. While both approaches produce data, neither is integrated with the biobank, which adds time, cost, and operational complexity before meaningful research can even begin. In-house sequencing requires building or accessing NGS infrastructure, selecting panels, performing library preparation, and analyzing data internally. These operational demands are resource-intensive and often divert time and expertise away from the scientific questions the study is meant to answer. Outsourcing sequencing introduces a different set of challenges. Depending on the vendor, they may only offer a small list of genes or genetic variations. Depending on the biomarker, this can add additional cost for more sequencing. And because third-party vendors operate independently of the biobank, researchers have to function as the middleman and must manage logistics such as sample transfer, chain of custody, timelines, and communication across all the parties. In both cases, sequencing becomes a bottleneck, adding time, cost, and operational complexity to the project. Because discovery integrates biospecimen access and NGS sequencing under one roof, pre-sequenced biospecimens offer a different approach altogether. In the conventional model, the researcher had to enter the workflow at the very beginning. Once a request was made, this is when all of the work started. Estimating frequency of the biomarker, pulling large numbers of samples, and sequencing them all to find the right positives and negatives for a given biomarker. From the researcher's perspective, this meant waiting weeks for all of the sequencing to happen, not to mention the costs. With SpecimenSeq, samples are molecularly characterized in advance, allowing the researcher to enter the workflow much later than before. Because the biomarker information already exists, researchers can immediately identify and access the samples they need. So instead of triggering a long screening process, when the researcher puts in a request, it is a simple matter of searching the database to identify the samples of interest from the biobank. This significantly reduces both turnaround time and cost for a researcher to get the biospecimens they need for their project. As oncology research became increasingly biomarker-driven, Discovery recognized early that pairing biospecimens with molecular data could significantly reduce discovery and development timelines. Rather than treating sequencing as a downstream step, we saw an opportunity to integrate it directly into the biobank model. In 2015, Discovery became the first organization to systematically pair biospecimens with genomic data at scale, transforming a traditional biobank inventory into a molecularly characterized resource. This served as the foundation for what became SpecimenSeq, and a fundamentally different approach to enabling biomarker-driven research. The timeline shown here highlights key SpecimenSeq milestones and product launches that expanded the depth, scale, and accessibility of molecular data over time. In 2015, we launched FFPE specimens with point mutation data generated by PCR, evaluating clinically relevant biomarkers like EGFR, KRAS, BRAF. In January 2021, we expanded to matched FFPE, double-spin plasma, and Buffy-coat trios with a targeted NGS panel, evaluating more than 70 biomarkers that included both SNV's indels as well as gene fusions. Later that year, in August 2021, we introduced SpecimenSeq DTCs, which are sequenced with that same targeted NGS panel. By October 2021, we further scaled our capabilities with standalone FFPE, now with an expanded targeted NGS panel evaluating over 500 biomarkers and adding copy number variants, tumor mutation burden, and microsatellite instability evaluation. In 2023, we introduced matched bone marrow BMMC and peripheral blood PBMC pairs with a hematologic oncology specific panel, enabling deeper insight into blood-based malignancies like AML. Most recently, in 2025, we expanded into tumoroids, characterized with whole exome sequencing and RNA-seq, supporting more advanced functional and multi-omic research. As the leading provider of pre-sequenced biospecimens, Discovery enables rapid identification of clinically relevant mutations, validation of therapeutic targets, and assembly of genetically defined cohorts without the time and expense of de novo sequencing. With curated molecular data already in place, researchers can accelerate cohort design, biomarker discovery, and translational studies with greater precision and confidence. We continue to actively grow our offerings through expanded gene coverage across multiple cancer indications, increasing availability of matched sample sets, and a dynamic inventory model that scales with research needs. Today, our sequencing operations support a run rate of approximately 1,500 new SpecimenSeq patients per year, with continued expansion planned. SpecimenSeq combines scale with deep molecular characterization across a wide range of clinically relevant alterations. The inventory includes over 1,200 clinically significant variants, spanning point mutations, gene fusions, copy number changes, and composite biomarkers like TMB and MSI. Matched set samples from the same donor provide important biological context. And importantly, SpecimenSeq is not static. Ongoing sequencing continually expands the inventory available to support a diverse range of biomarker-driven precision oncology programs. You can explore all the genes and genetic variants available through SpecimenSeq on our website.